Hello, and thanks for coming to our poster presentation on improving long-read consensus sequencing accuracy with deep learning. My name is Avantika Lal, and I'm a senior scientist on the genomics team at NVIDIA. And this work is a collaboration with our colleagues at PacBio. We are excited to share our preliminary research showing that deep learning can improve the accuracy of PacBio's HiFi sequencing technology. The PacBio HiFi sequencing method is based on single molecule sequencing of a circularized DNA molecule in repeated passes, producing multiple subreads which are individually about 90% accurate. From these subreads, a consensus sequence is generated using the CCS algorithm. These consensus reads or HiFi reads are long as well as highly accurate. However, they still retain some errors, predominantly insertion and deletion errors at homopolymer regions. Our goal is to use deep learning to increase the accuracy of HiFi reads even further. To do this, we take each read and generate a pileup by aligning all the subread sequences to the HiFi read sequence. For each column of the pileup, we then count the number of occurrences of each base, A, G, C, T, or no base, on both the forward strand as well as the reverse strand. In addition, we encode the consensus base at each position and the base quality score for that consensus base. We pass each encoded read through a deep learning model consisting of multiple convolutional layers, followed by a bidirectional GRU layer that integrates long-range information. For each position in the pileup, the model predicts the most likely correct base at that position, and these predictions are then stitched together into a polished or corrected HiFi read sequence. We tested our deep learning strategy on a human genome, that is an HG002 dataset, sequenced on a Sequel II platform. We first aligned all the HiFi reads in this dataset to the human reference genome and selected only those reads that aligned within the benchmark regions defined by Genome in a Bottle. Further, we selected reads that didn't overlap with any benchmarked variants within these regions. We did this to eliminate genomic variation from the dataset, so that any remaining differences between the read and the reference sequence could be assumed to be sequencing error. So for each selected read, we took the subsequence of the human reference genome to which it aligned as the true sequence, and any differences between that sequence and the read were considered to be sequencing errors. We split the remaining reads by chromosome and trained a deep learning model using the reads from one set of chromosomes and tested the model on the reads from another set of chromosomes. In total, there were 3,586 HiFi reads in the test set. We found that the deep learning model reduced the number of sequencing errors in the test set by 29.6%. In other words, it increased the average read accuracy from 99.83% to 99.88%. We can also represent read accuracies as QV scores, which are calculated as minus 10 times log 10 of the probability of an error in the read. Over here, we plot the QV read accuracy for the HiFi reads with different numbers of subreads before and after they were polished with deep learning. And we see that deep learning consistently improved the accuracy of these reads. If we break down the sequencing errors by type, we see that deletion errors in the test set were reduced by 20%, and insertion errors were reduced by 36%. Over here, we show the number of insertion and deletion errors in the test set before and after polishing, broken down by the length of the homopolymer at which they occur. Next, we wanted to test whether this model trained on human data can generalize to different datasets and different species. And so we applied the same model to 10,000 reads from an E. coli dataset, which was also sequenced on the Sequel II system. We found that the model trained on human data reduced errors in the E. coli reads by 26.2% and increased the average read accuracy from 99.75% to 99.815%. However, Although insertion errors in the test set were reduced by 43%, we observed that deletion errors were increased slightly by 8%, suggesting that further work is needed to improve generalization. In conclusion, we've shown that deep learning can improve the accuracy of PacBio hi-fi sequencing. In our experiments, we've seen that our current models, although they are trained on a relatively small number of reads, still reduce the overall error rate by 25 to 40%. And the models are particularly effective at reducing insertion errors that increase homopolymer length, as well as long stretches of errors in the read sequence. We also see that a model trained on human data could generalize to a different dataset from a different species. We believe that these initial results can be improved by training on more hi-fi reads from a larger set of regions in the genome, and by training on data from more diverse sources. It'll also be interesting to see whether this improved read accuracy can enable more accurate genome assemblies and variant calling. Improved read accuracy may also reduce the cost of hi-fi sequencing by enabling high-quality consensus reads to be obtained from fewer sequencing passes. And it may also improve the yield by allowing low-accuracy reads that were previously discarded to be retained. For more details, please check out our recent preprint on bioRxiv, and feel free to reach out to us via the conference platform or on email. Thank you.